All right. Hi, everybody. Welcome to the March 2023 edition of Cells to Society. I'm Andrew May with UVM Extension Center for Sustainable Agriculture, and I help to coordinate the series along with Chris Callahan and Hans Estrin. So today our topic is microbiology and molecular genetics. We're excited to have two great speakers with us today. The first presentation is going to be bacteria in our water system with Matt Wargo, who is the Associate Professor of Microbiology and Molecular Genetics at UVM, and RNA Viruses in Nature with Emily Bruce, Assistant Professor in the Department of Microbiology and Molecular Genetics, also at UVM. So a little bit more about Cells to Society seminars. Our overall goal is to promote connections, transfer of knowledge, transdisciplinary collaboration, effective leveraging of research findings, identifying new research questions, and collective impact. So this seminar series is meant to be an opportunity to promote collaboration and ultimately to weave a stronger network of relationships and professional connections. So this is our team currently putting the seminars together. So again, thanks to Chris and Hans, as well as Kathy and the rest of the media team. Our effort is entirely collaborative. So we definitely appreciate any suggestions, which you can submit at the link at the bottom of the slide, go.uvm.edu/cells to society seminar. And you can also view the schedule and past recordings there as well. So the way that it works is that we have monthly virtual seminars. The seminars, including the one you're seeing today, is designed to be an opportunity to share research, education, and work within CALS and beyond. So these seminars typically have 2 speakers each. And then we have quarterly out-of-the-box brainstorming sessions, which happen every 3 months or after every 2 seminars, where we select multiple emerging themes for potential new projects and discuss them in small groups. So this is a much more engaged and interactive collaborative around themes and project brainstorming, and hopefully can result in some good grant proposals as well. So our standing schedule is every second Thursday at 2 p.m., which will occasionally change, like today. So we're changing the time today, but so an example of next month's out of the box workshop, we will be announcing some of those topics in the coming weeks. So again, if you have any suggestions, please submit them at the link shown at the bottom of the screen. And finally, as we've mentioned, please visit the Cells to Society website seen here for more information on upcoming seminars and links to recordings of past seminars. Right. So without any further ado, I'll hand it over to you, Matt. Looks like you might be muted, Matt.
Ah, there we go. Okay. So you're seeing slides here.
Yeah.
Okay. So so thanks for having me here today. And I'm going to talk about bacteria and water systems and kind of, go into our work, but kind of give you definitions, what I mean by the bacteria and what I'm talking about when I'm referring to water systems. And so if we count the number of cells on the planet, this is a pie chart of all the cellular life on the planet. All of them essentially based on this graph are bacteria, right? And that's because bacteria are very small, but there are also a lot of them, 10 to the 30th or so on the planet. So they're everywhere and they outnumber everything else. But if we think about this in a biomass kind of way, we see that plants obviously sort of dominate biomass. And then most of these bacteria, or a good chunk of these bacteria, are photosynthetic. So they're also primary producers. But you see even the animals here are this little sliver in terms of mass of life on Earth. And so bacteria contribute in both number and mass, and they're everywhere. And one of the things to take away from today is we're never going to get rid of them in any system that has people and animals and plants. They're always going to be around. And so how do we understand them and how do we control them? If we're talking about water systems, there's both natural and plumbed and human-based systems. And I'm really focused on the plumbing-based systems, or the plumb systems, so both wells and municipal water systems. but also what happens to that water after it comes out of those sources. The microbiology of natural water systems is very different than that of human controlled or plumbed systems. So famously, John Cleese asked, what have the Romans ever done for us? And aqueducts, right, was one of the answers in Life of Brian. And so, but what they probably did was give us the first exposure to plumbing pathogens, and as the first sort of large scale multi-city kind of water system. So what do I mean by plumbing pathogens? Well, there's two real types of pathogens. There are what are officially called the opportunistic premise plumbing pathogens, or the OPPPs. Those are the ones that live in the system that we're never going to fully get rid of because they can live their entire sort of lifestyle in that system. That's the contrast to the waterborne pathogens, which are due to contamination. And so if we think about things like E. coli and other enteric pathogens, as well as Listeria, if they're in the water system, they got there from somewhere, and there is contamination that you could clean up to remove them. Whereas these, including Legionella, non-tuberculous Mycobacterium, a number of other bacteria, you're never really going to get rid of because that's a niche that they live in. It's not usually their only niche. A lot of these are also soil bacterium or natural water bacterium, but they can do well in our plumbing systems. And one of the reasons is because the plumbing system isn't just perfectly clean, copper or stainless steel or PVC piping. It's all the stuff that is associated with that. So these biofilms on the inlines, this is a copper pipe for inline fitting, or even more sort of the drain of a water system, because especially, if you think about dumping pasta water or something down the drain, you're giving a lot more food on the outflow than those bacteria have on the inflow. Now, one of the reasons you get this amount of biomass in something that you consider not a nutrient source, right? Water, we think of potable water as pretty clean, and it really is in the US. But there are a lot of dissolved nutrients that the bacteria can still take advantage of. And so they're not growing rapidly, but they're given constant flow of water, or at least intermittent flow of water, and they can use that to grow. And they can also strip metal from here and cause, metal corrosion. And so the other thing to think about in these systems is that they're always polymicrobial. There's not just one bacterium, and not just, typically 10 or 20 bacterium. We're talking usually hundreds of bacteria in this system. And so I mostly study lung infections, and my real interest is, actually, if we go back to this list, is things like Pseudomonas aeruginosa and Stenotrophomonas multifida, which are really important lung pathogens in humans, and humans typically get them from the water system. So not everybody can get these. You have to have underlying conditions, essentially, or immune suppression to get them. But when we get them, they often come from the water systems. So one of my interests is essentially what they're doing in the water system before they get into the human. But really, this was driven by bacteria from space. And our model water system turns out to be from the International Space Station. It's from the Potable Water Reclamation System. That's the machine that has a sticker on it that says "Making today's coffee, tomorrow's coffee," because it's actually the urine recycling system. But the bacteria on the outflow of that system are very similar to all the bacteria that we'd find in any tap water that we turn pretty much anywhere on the planet. But the only trick is, in the NASA system, they actually get a lot fewer nutrients, a lot fewer compounds coming through in that water than we have here, because it turns out NASA's expectations for water purity are much higher on the space station than the EPA's recommendations for Earth-based systems. They have much lower tolerance for that. So these bacteria are because they were on the space station and because they were sort of kind of stripped down and they have fewer nutrients, it's actually a simpler community. Probably about 40 species comprise maybe 90% of the biomass in that system. And we further stripped it down to use it as a model. And one of the benefits is it's colorful. So hopefully this shows up on your screens, but there are some yellow and this one's even more orange. There's a pink version, a slightly greenish bacterium. So they're not just beige like most bacteria are when you streak them on plates. And we can view their biofilm formation, whether it's on sort of pre-filter material here or in flow biofilms where we're sort of mimicking pipe flow and visualizing the bacteria. In this case, all the bacteria are stained in red. And then if you look in green is one species and purple is a different species. So not everybody is sort of intermixed everywhere. This is actually a spatially and species composition-wise heterogeneous community. And one of the things that we want to understand, even sort of disregarding infection, is how these members of these communities interact to make the biofilm a stable, robust kind of system. And there's a lot of data on this slide, I apologize. But what we do is we measure biofilm by looking at essentially the air-liquid interface of culture. So the culture liquid goes from the bottom here to the air-liquid interface. And we grow the bacteria, not shaking, let them stick. They generally stick at the air-liquid interface and sometimes down a little further. And then we take the bacteria out, we wash that actually fairly rigorously, and then we stain it with crystal violet to have it show up better. Crystal violet, if you've ever worked with it, stains every type of living material and pretty much any fabric as well. And here, if we take our 6-member community that we have, our tap water community, we form biofilms and we can remove some members and not really impact the biofilm at all. But what's shown here on this graph and what's shown here in this well is if we remove these 2 specific species, we essentially completely ablate biofilms or remove it. This is log scale, so down to like 4% or so of the actual amount. This then tells us, well, those 2 species they're like the keystones. They're the critical factors requiring biofilms. And it turns out that the Chrysobacterium part of this, the CG, needs another member as well to interact with, and that's Ralstonia. And I bring this one up because I'm going to talk about it in terms of Listeria here in a minute. All this slide really shows is that Ralstonia and Chrysobacterium, when together, in this case, versus either by itself, form a much stronger biofilm. I mean, it's not due to necessarily more cells total in the culture. It's due to the fact that they stick a lot better. And so if sticking to a surface is an important part of the biofilms, especially in flow systems. And so understanding how that works and if we could manipulate that in any way is part of what we're interested in. One of the things that that links back to the health part is that these opportunistic plumbing pathogens have to live in the same environment as all these other bacteria in the water system. And so how do they do that? Do they do well against those members, or are they generally outcompeted? And so we can set up systems in the lab to kind of study that, where if we, in our mimic of tap water, if we put Pseudomonas in there alone, which is a pathogen that we're interested in, if we put Pseudomonas alone, it grows to almost 10 to the 7th cells, which is a lot considering how little amount of nutrients there is in this system. Although that's still below what you can see with your eye. So that would look perfectly drinkable to you if it were actual water that you would drink. However, if we mix it with the full 6-member community or the community missing any single member, so all these are 5-member communities missing that individual member, we see that there's 100-fold less Pseudomonas in those conditions. So it's outcompeted to some extent. And then we can mix Pseudomonas together with individual species and see that certain species are antagonistic to Pseudomonas by themselves, especially Sphingomonas and Ralstonia. So understanding how these bacteria in the water system impact these pathogens, is something we're interested in, and especially if, for instance, we could promote one of them to decrease the ability of that water system to carry a pathogen, would be interesting. So that's the sort of the potable water system, the premise plumbing, the what comes out of your faucet or out of your shower head. But once it leaves the shower head in the faucet, we're still doing a lot of things with that water, right? And if we think about it in terms of the food systems, we're using water for processing, and the water's coming out of this, but then it's running across the floor and down the drain, and there's splashes from the floor. We're washing vegetables or growing vegetables with water, especially in completely open systems. We worry about sort of pathogens and what's growing in there. And I really got linked to the food side of this in collaboration with Andrea Etter. And it was really spurred along by Sophie Unger, who was a food surf undergraduate awardee last summer. And she was looking at how Listeria and Ralstonia can form together a much stronger biofilm than either of them can form themselves. So Listeria is this enteric pathogen that can also cause meningitis and issues during pregnancy. It's a really important pathogen, especially in dairy. And so there was data out there that people could show that some species of, or one particular species of Listeria or strain of Listeria could interact with one strain of Ralstonia. And what we wanted to do is, for those of you who don't think about strains in bacteria, these are usually genetically different to some extent and often have very different behaviors between strains than you would expect from such a simple organism, but from things that are not genetically too dissimilar. And so a lot of times what we study in a lab strain is not necessarily applicable to the natural environment, the strains that exist there. So what Sophie was doing is looking at, these are again, these 9612 biofilm plates, flexible PVC plastic, that none of these three strains, this is supposed to be 19, this middle one. None of these three strains of Listeria are capable of forming much biofilm by themselves, and this Ralstonia from the space station doesn't form much biofilm itself. But if you mix any of these three Listeria with the Ralstonia, we get much more biofilm formed. Now, this wasn't, other people had shown that this could happen, but being able to show that it happens over many strains, and now Sophie has done 8 strains of Listeria and 8 strains of Ralstonia, and showed that every possible interaction, we have a promotion of biofilm. So this seems to be a fairly universal thing. We're going to increase the strain number. Unfortunately, after Sophie leaves, because she's going to graduate here soon. And if you look at those biofilms, zoom in, what is this, 4,000 or 4,300 times in these two pictures. This is scanning electron micrograph showing the mixed biofilm of Listeria and Ralstonia sort of clumped together. And these are fairly large, robust clumps. I don't know if those of you who were at Andrea Etter's last Cells to Society, I think the last session, her Listeria biofilms were really sparse. And when I first showed her this EM, she was just aghast at how much biofilm forms on the surface. And it's really robust. It's actually pretty hard to remove, even from this plastic surface. In this system, they seem to be mixed together and in contact. But interestingly, it doesn't require contact. So one group, actually now a little while ago, showed that the supernatant of Ralstonia can make Listeria form these clumps, even if they're not attached to a surface. They'll just sort of spontaneously aggregate. And if you have them mixed together, you get the listeria, which are these darker cells. This is a cross-sectional electron micrograph. Or the listeria aggregate around the Ralstonia. And so it's sort of there is an interesting interaction, and one of the things Andrea and I are interested in is who's driving the interaction and how are they doing it, and can we tell if a place is going to be a risk for repeated listeria exposure if they also have a lot of Ralstonia. So that's one kind of question. And that's really the where are we going part of this question, which is so what are we going to do with this information, and sort of how to more broadly how does it impact us? And from the plumbing pathogen side, we are interested in how these two bacteria specifically suppress Pseudomonas aeruginosa in water systems. And Catherine Armbruster is down right now. She's finishing up her postdoc at Dartmouth, but she'll be starting her lab at Carnegie Mellon fairly soon. So her and I are going to be collaborating on this because she has a lot of experience through the CDC with their water system analysis, and we're going to look at that. And then with waterborne pathogens, with Andrea Etter, as I was mentioning, digging further into this Ralstonia listeria interaction. And if you look at the literature and you look at sort of experiments that we've done that I haven't shown you here, Ralstonia really seems to be able to promote biofilms from quite a large number of potential pathogens. So the question is, do the pathogens really need Ralstonia in the system to be infectious out of that system, for instance? If we were able to remove Ralstonia in a targeted way, could we decrease the ability of that water system to carry enteric pathogens, to carry listeria? So these are the kind of things that we're going after. And obviously, I do very little of this work. It's been done by lots of other people. These are just the historical. Currently, we have just two grad students, my lab manager, Kristen, and then Sophie Unger, who's listed down here and from biochemistry in the lab, but collaborations across campus and elsewhere. And the funding, all the water work, as you can imagine, the International Space Station stuff is funded primarily by NASA, but it's also has been funded by the Cystic Fibrosis Foundation and the NIH, because when those community members are going to impact health, that's partly coming from there. So with that, I will end the show here and be happy to take any questions.
Fantastic. Thanks, Matt.
Yeah. But let me find you guys. Stop sharing.
So let's well, I noticed a few questions in the chat. I think we can maybe move on to Emily's presentation, if that's all right, and then we can think about some questions for both of you all at the end, if that's okay. Great.
And I think I need to be given permission to share.
All right. Just 1 second.
No worries. I'm not normally a Teams person, so I was trying to figure out. Button's grayed out. Ah, there we go.
That's all right. Yeah, there we go. All right.
No, I'm not going to upgrade the audio driver. All right, can you see this presentation?
Yeah. Mm-hmm.
Okay, let's see if I can. All right, and are you seeing the full screen version?
Yep, it looks great.
All right, so the story I'm going to tell you about today, I'm going to give just a very brief disclaimer up front. The findings that I'm going to show today are confidential and they're not for onwards distribution, which primarily means please don't share them with journalists, which sounds odd, but is actually a problem I've had with this project. And the reason for that is we want to have all of the data uploaded in a manuscript that's available for everyone in the public to see on a preprint server before we talk to the press about it, because we found that that's the best responsible thing to do with COVID data in the pandemic is to make sure that the data is available for other people to look at before we talk about it publicly. And that doesn't apply, of course, when we're talking internally with internal presentations and academic audiences and so on. But the project is about the prevalence of SARS-CoV-2 in Vermont wildlife. And the reason that this is an issue is that right here in North America, there have been a number of studies showing that the virus that causes COVID-19 in people, which is SARS-CoV-2, is also able to infect a range of other mammals, including white-tailed deer. And this, I think, was surprising to many people. But there have been a number of studies now that have shown throughout North America that deer can become infected with the virus that causes COVID-19. They can spread it between themselves, and they can also infect people again in what's called a zoonotic spillover, or a introduction and spill back event. And the reason that people are concerned about this is that when viruses jump from one species to another, you can select for the presence of different mutations in how the virus replicates in a different species, and that can introduce new properties to the virus, which might be kind of bluntly bad news for people if it comes back. And these can be things like viruses evolving to use or bind to slightly different surface proteins, which might mean that people who'd been exposed to the original strain wouldn't be able to neutralize the new variant as well, which is something we've seen happen purely among people in the way that the virus has changed. And so this is a thing that in general is something that a lot of virologists and people in this kind of pandemic preparedness space want to keep an eye on. And this is a project that started here at UVM in a collaboration between me and Brittany Mosher. And you can see from this tweet here on the right, I didn't know Brittany really before I saw her tweet, and she was talking about having resources to look at COVID modeling. And so I reached out and I said we should grab coffee, and I said, you know what, if you're interested in doing anything on the wet lab side, this is what my lab does. So I'm a virologist, and we do basic wet lab work at BSL-2 and BSL-3 with SARS-CoV-2, and so maybe there's an interesting way we could collaborate. And so here's just a couple of the articles that had come out recently showing the detection of multiple different variants on multiple different occasions of SARS-CoV-2 in white-tailed deer throughout North America. And this, people have detected multiple different variants of SARS-CoV-2, and this is a thing that has happened. Basically, it's not a one-off coincidence, but that this has happened in North America and also on the Canadian border right across the northern border here. And you can see from these titles, these are viruses that are acquiring genetic changes that differentiate them somewhat from the viruses that are well suited to infect people. People have also seen variants of concern that are extinct in the human population, things like alpha and gamma, that were circulating in deer for months after they had been they had ceased to circulate in the human population. And so that again raises questions about what viruses are circulating in deer and the extent of what is actually going on. And the primary kind of questions to come out of this project, or what spurred it from the beginning, are whether white-tailed deer serve as a reservoir for SARS-CoV-2, which I think the evidence is indicating now that they really can. What are the likely pathways of transmission between deer and between people? And the larger question of how the risk can be minimized of SARS-CoV-2 becoming endemic in white-tailed deer. And I'll say, just a full disclaimer up front, I'm a basic scientist who works on viruses and how they infect cells and how they do the things that viruses need to do on the smallest of scales. I'm not a policy person, and I'm not someone who's poised to answer many of these big picture questions, but I think that we're in the position to provide some of the basic information that can be useful in this larger space. And as I mentioned, this is a project that was really a huge collaborative effort led between me and Brittany Mosher. There's a postdoc in her lab, Elias Rosenblatt, who's also involved, and Brittany's involved in a much larger project looking at modeling in the presence of SARS-CoV-2 in deer that really involves many of these other institutional partners. The piece that we were directly working on with surveilling these Vermont deer was led by her and her access to people at the Vermont Department of Fish and Wildlife, the UVM Wildlife and Fisheries Society, which is an undergraduate club, and it was the undergraduate club members that did the majority of the sampling that we did in this study. And on my side, it's really been spearheaded by a fantastic graduate student in my lab, Hannah Dupre, with the assistance of other lab members. And then Margaret Mills at the University of Washington in clinical virology did all of the PCR testing of all of these samples, which numbered in the well over 500. And so that's at the scale where you really need the automated robotic systems to be able to process these samples. And so Margaret is one of the lead clinical virologists there and has access to all of the CLIA certified testing space where they perform the majority of the tests for COVID-19 in the Pacific Northwest. And so this was a great group, and we kind of all combined together with our relative expertises and saw the opportunity to answer a question here about what was happening in Vermont wildlife, because I showed lots of other papers from other Northeast states and across the Canadian border, but there is no currently available information on the presence of SARS-CoV-2 in Vermont wildlife. So the way we set up this study, we collected samples from either trapped or hunted animals in Vermont. So for the trapped animals, these are typically fur-bearing animals that are trapped for pelts. When you trap an animal in Vermont, you're required to give the carcass to the state for various wildlife biology testing. And so we had access to these carcasses, including coyotes, otters, foxes, bobcats, fishers. I think there might have been some mink, as well as hunted animals. And so these were deer and a couple of bear. And we collected these, the deer samples on the 2 opening weekends of deer season. So in October, there's opening season for youth weekend, and then in November, there's the opening season for rifle season. And so we had pairs of undergraduates and students and technicians in my lab who spread out really all across the state and staffed these checkpoints where hunters bring in their deer after they have their hunting license, they shoot their deer, they bring the animal into the check station, and there's various measurements that are taken by state wildlife biologists, and we piggybacked on those collection sites to collect COVID swabs from the noses of these animals, basically exactly the way you do your COVID swab yourself during the middle of the pandemic. This is what we did with these deer carcasses, and then we're able to store those in PBS at -80, so they're stable and long-term freezing. And then we aliquoted these samples and sent aliquots to Margaret at the University of Washington, where she extracted RNA using a robotic platform, and then performed RT-PCR to look for the presence of viral RNA specific to SARS-CoV-2. And she used a couple of different primer probe sets. These are really well-validated assays that are very, very specific to exactly the SARS-CoV-2 virus. And because they are a clinical lab, they have this whole pathway set up with controls to ensure that each sample we collect is able to amplify and that there's nothing that is inhibiting that reaction, and controls on each plate of low-level positive samples to ensure that the assay is working as we expect on each plate. And these are standard controls in the field, but the University of Washington really are the experts in this kind of work. They do a lot of this viral pathogen discovery for USAID as well. So they're leaders in this space. Let's see. Sorry. All right. I'm using different keys in Teams than in Zoom, so that's exciting. All right, so here are some pictures of the sample harvest of people in my team doing these collections, and yeah, apparently each time the hunters came up, everybody always joked that, what are you doing, swabbing the deer for COVID? And yes, indeed, we were swabbing the deer for COVID. And so we ended up with, well over 500 samples. So the 2021 season was more of a pilot season for deer. Brittany and I really got together just barely in time to get the paperwork sorted for a very small pilot collection with deer, where we went to a single checkpoint in Jericho on opening weekend. And so we got 18 deer from the 2021 season. But Hannah was able to collect samples from well over 100 fur-bearing animals. And you can see here on the season 1 map, you have the circle showing the geographic distribution of where animals were collected throughout the state, and the size of the circle indicates the number of animals that were collected. And then here in season 2, you can see we were able to collect more in the way of cases. And particularly that was because of the large number of deer that we got in season 2. But we processed the samples for 2021 first, and we found no detectable COVID. And so we were thinking, or SARS-CoV-2, we were thinking that we might find things in season 2, but we did not find anything in season 2 as well. So we swabbed 470 deer, as well as 3 bears. I think maybe we lost one of the bear samples. We ended up with 2 in the final set. But we didn't detect any COVID in either of these populations, which was a little bit surprising to us. But we are quite sure of these results with the internal controls that we have in our lab assays. There are a couple of caveats to that, which I'll talk about at the end. But I think one of the explanations that this could be is that if you look at the number of COVID cases in Vermont, that's shown here with the yellow line, and here's the time. So this is from 2020 out through just about the end of 2022. You can see this blue square here is when we collected samples from fur bearers, and that does somewhat overlap with the primary case peak in Vermont. You can see the 2021 hunting season for deer, really there had been very little circulation of COVID in Vermont at this point, which was particularly notable compared to other states in the US. Vermont controlled the COVID outbreak very tightly for a very long time. And so it's possible that we're not seeing any COVID here early on because it simply hasn't had the chance to spread from people into deer yet. You can see that the 2 sampling weekends here in pink at the end of 2022 are after this peak of COVID introduction in January of 2022, but we're still not detecting active infection in deer. And it's also worth noting that Vermont had relatively less in the way of COVID. And you can see the distribution here. This is, we've got Vermont on the left and Vermont on the right and New York on the left. And on this left panel here, we just have the raw number of case counts. So you can see the areas in yellow have a high total number of cases. If you look on the right, this is plotted against the number of people in each county. So you can see the prevalence. You can see in either case, Vermont has low amounts of COVID compared to New York, particularly kind of Western New York and this area down here with New York City. So I think it's plausible that one of the reasons that we're not seeing it here in Vermont yet is that there is less COVID-19 in the people that the deer have the chance to interact with. And if you look here, this is a beautiful paper that just came out. Sorry, I don't have the citation on here, which I should. And if anyone's interested about it, I can send it to you. But it was a lovely paper that just came out looking at the prevalence of SARS-CoV-2 in deer in New York State. And here is season 1, which I believe is the 2020 to 2021 season. And here you see a little bit of COVID early on. And this is also in those places that were yellow in the human COVID maps. And then you see much more spread in the second season. And they also showed nice evidence that this was spreading in between animals within the state, and nice controls to show that these were not false positive results. And so I mentioned that I would talk a little bit about the caveats too of our work. We exclusively took nasal swabs of animals and looked for the presence of viral RNA. And that, the thinking is, would measure the presence of active infection. Other studies have taken other kinds of samples. They've taken a lymph node in around the neck area, and they have looked for the presence of viral RNA in that lymph node. And some of those studies have reported higher levels. It's possible that there's higher levels of viral replication in that lymph node, and that's why they see higher levels. I suspect that that's not the case. I suspect it's the case that they are simply sampling in a place where there is more COVID. The reason we did not collect that lymph node is that is the tissue type that's harvested for surveillance of chronic wasting disease. So it's piggybacking on another surveillance effort that happens in many places. There is not any surveillance for chronic wasting disease in Vermont at this time. We haven't found it. It's not considered as much of an issue, and New York State has an excellent surveillance program. So we kind of piggyback on that, and not having the resources to do that ourselves. So we did not have access to that lymph node because, yes, I was not going to train 50 undergraduates to start doing dissections in the necks of deer to find this lymph node. Other people have also taken dried blood spots to look for the presence of antibodies against the virus, which could show the presence of a prior infection, as opposed to looking for the active infection, which is what looking for RNA in the nose is. So those are some of the distinctions. But I think even if you compare the people who looked in the same way that we looked, in places with either more human COVID or potentially more farmed deer facilities or interactions between people and deer, they found higher levels of COVID. So I think if you were to ask me to predict, I would say that we would probably start to detect this in Vermont in the next year or two, but it's not currently circulating at high levels in deer, at least as of November of this year. And that's I think quite possibly a reflection of the fact that there was less COVID-19 in the Vermont human population during this time frame, as well as the fact, that should say there's no farm deer facilities in Vermont. There are farm deer facilities in New York. So I think that is another possibility, that there are more interactions between people and deer in New York, and that is one of the things that helps the virus spread more quickly, is having the opportunity to jump from one species to another by having those types of closer interactions. And this is something that people have seen in all kinds of farmed species. Minks are a particular concern, not just for SARS-CoV-2, but also, as we've seen in the current avian influenza outbreak, they're a species that has respiratory tracts that are similar to people, and when they're farmed, they're kept in very close proximity to each other, which has the ability to magnify viral introductions from one species and then back into people. So that just a side comment on farmed mink. And I put all of the acknowledgements up front. So again, this was a very, very collaborative effort. And I will stop a little bit early and just give time for questions for both of the presentations. So thank you guys so much.
Fantastic. Thank you. So we do have a little less than 10 minutes for questions. I know that there are a few in the chat so far, definitely for both Emily and for Matt. So maybe, since we just heard Emily's presentation, maybe we can address Matt's question here. Is there a good correlation with deer density and positivity rates? Much less dense deer population here than in MA, Connecticut, New Jersey, PA, or a deer person contact rate of some way? And that question comes from Matt.
Yeah, that's a great idea, Matt. Hannah's putting together the figures for this paper now and figuring out how to use GIS to display all of these things. So I actually don't have an answer for you, but I'm going to write that down and I'm going to ask her to make the figures so that we can get it into the preprint. I do know from her paper that Vermont has a 1-to-5 ratio of deer to people. So there's, but I think that I don't know off the top of my head how that ratio compares to other states. So let me check in with her.
So switch over to Matt, maybe a question from Hans. Hans, do you want to just go ahead and ask your first question about biomanagement and listeria and microdairy processing?
Yeah, just, I mean, this kind of comes from, thanks, it kind of comes from last time, and just thinking how prevalent, or if listeria kind of comes into these systems, especially the microdairies in Vermont, etc. So just, testing that in situ, how would, like, paint a picture of how that could look, if it just, or like speculate on, I heard you mention that that would be really cool to figure this, or that you're working on this, obviously,
Yeah.
but what does that look like in the real world or down in the trenches?
In the real world, for selective removal, you're pretty much talking about bacteriophage, which are the bacteria that infect, or the viruses that infect bacteria. And so, and NASA was super interested in bacteriophage treatment because they don't need serious approval to move it up to the space station. They can't use bleach or ethanol for safety reasons up there, but bacteriophage are pretty safe. They could bring them up and flush them. The trick is, once the biofilm was established, the phage would not kill a substantial enough number of the population to impact the biofilm. If we added them before the biofilm formed, we could show that they were really efficacious, but that's like never, like, I mean, maybe if you were just starting up your cheese facility and you could like swamp them in phage, but other than that, yeah. So I think it's hard to figure out how to selectively go in there, and people have talked about there are predatory bacteria, like bacteria that eat other bacteria, and trying to use those. But yeah, it's a great question. If we had, a lot of these are also very drug resistant, so to antibiotics and to biocides, and so it becomes hard in that way to if you could think about, treating them with a drug to remove some of them.
That, in what you mentioned before, I think that is a possibility, because, at least in Vermont, I run across microdairies starting up, and they're, in construction phase, and there's projects, and frankly, in the produce world in general, not just processing, we help consult on tons of, new wash pack stations, and so it's like new construction, and that might be a kind of cool, I don't know if it's something that, I'm sure growers or people might be interested in at least trying some of this stuff if it wouldn't impact their business in a negative way.
Yeah.
Looks like Andrea has a question. Go right ahead.
You're on mute. Wait, you're muted, Andrea.
Sorry, it's actually not a question. It was just more of a follow-on comment to Hans' question. One thing that dairies can do, and I have a project working with a dairy on this right now, is to see if some lactobacillus will inhibit listeria. We're not entirely sure how they do it, but they do. So that's an option. I don't know how well that works with biofilm, but it's definitely an option. And Emily, I loved your study. As per my comment in the chat, I think these things always end up surprising us. It's just never what you think it's going to be.
No, we were all ready to sequence things and look at the differences between them, and to isolate viruses in the BSL-3 and look at how the properties of things changed, and then we got nothing at all. We're debating if we'll try to do this for the next season. It's really Hannah's brainchild that she really wants to see to the end, but well, yeah, The other thing I've learned from this is that there's no small project. There's no like small side thing that you're just like, let us do this. It'll be easy. This is what I thought at the beginning. I was like, this is easy. We can do all these things. Why wouldn't we want to look? It's so curious. But there's no small project.
Especially not if you're doing field sampling.
Or getting something published, just getting it
True.
through review. It's like a million hours of paperwork just to get it out there.
I have a quick question for Matt. Well, it might not be quick, and this is coming from a place of total ignorance beyond the 20 minutes of your presentation. But you mentioned the relationship of your work to food systems, and the first thing I thought of when you showed that picture as well was of kind of indoor aquaponic or hydroponic systems, and it made me think about the really controlled nutrient availability that was needed, and like the controlled temperature and the controlled pH that happens within all of those kind of tubes and piping that are working throughout and some of those systems are huge. There are systems in the country where it's just massive amounts of tubing that have these very controlled amounts of nutrients playing very controlled pH, very controlled temperature. So I guess I'm just curious if your work is branching into that area, and to what extent your conversation about the formation of biofilms is of concern to places like that, to some of these like larger indoor operations.
Yeah, I hadn't really thought about that at all until I don't know if it was the food systems like steering committee or something that I was on, or something else I saw with the food systems set up, which was how much hydroponics we actually do have, in Vermont, in some form or another. And yeah, all these things that I study would be in the hydroponic system, right? Because even if you're pulling it from, a non-municipal source originally, like you're pulling it from the river and lake or something, once you start getting it into into a tubing system, you're going to start selecting for these kinds of bacteria. And I think it would be fascinating to look at. And I think during that meeting, I was doing the Google searches, right, for like what had been done in hydroponic systems. And there's certainly Ralstonia in there, and how they impact the food safety. It's interesting, right? Because even for the plant, it's a completely different community they have to deal with than what's in the soil. And so even what they're sensing from their environment is completely different. So both from a safety perspective, but even from a taste perspective, because there are some of those ones where, you change the soil a little bit, or you change the bacteria that are associated with the soil, you get differences in like tomato taste. And does that happen in the hydroponic system? Do you get altered taste depending on what's infecting your water lines and that kind of stuff? It's a fascinating question, I think.
Truly, yeah. I mean, especially and for aquaponics as well, where you're actually developing like a culture of bacteria in the water system that's continuously cycled. So it's really fascinating to think about how the introduction of a different bacteria could compound and interrelate to other bacterias and create, a totally new environment that could be dangerous, I guess, and on one end of the spectrum, or just curious on the other end.
Yeah.
So let's see. So I actually think we only have about 2 minutes left, and I tend to try to stop these just a little bit early in case we end up going over, which has happened in the past. So to be respectful of people's time, I think we'll call it there and go ahead and stop the recording. But thank you so much, Emily and Matt. This has been really great. And I hope everyone enjoyed it as well. So again, for those that are listening and watching, go ahead and over to the Celsus Society website to see what's up next. We've got a lot of other great presentations this year as well. So thanks again to our speakers.
Thank you.
Thank you. Have a good day.